welcome to Sir Majesty Easy World Science channel, the easy world of science. Today, we're going to look at biochemistry of the nucleic acids. The biochemistry of the nucleic acid, that is the biochemistry of DNA and RNA. I'll get in details in terms of the structure, the constituents of it, what makes it up, and other information, even the base pairing rules, the complementary base pairing, and so on and so forth. So happy watching, and don't forget to ask your questions in the comment box. So we have so the acids in the nucleus are actually called nucleic acids. Remember, they are not restricted in the nucleus. They can be found outside the nucleus. So we have extrachromosomal DNA. So it's not going to be surprised if you see any of these acids outside the nucleus. But in nature, they are found in the nucleus of a cell. And remember, DNA is the major genetic material for every other living organism, except some sets of virus, the retroviral, the RNA viruses, where RNA carries the genetic information. First, we have two types of these nucleic acids. Nucleic acids are of two types. The RNA and the DNA. The DNA means deoxyribonucleic acid, and RNA means ribonucleic acid. I'm going to consider their biochemistry, their differences as I'm teaching. You will now know their differences, isolate their differences by yourself. But I may give you a brush down of some of the basic differences between them. At least they differ in their ribose sugar. In the pentose sugar, this one have one oxygen less in the pentose sugar, while this one have one oxygen more. Because of the one oxygen less, it is called deoxyribose. So it is deoxy. But both of them are actually franose structures. They occur in the cell as a kind of that's the sugars that makes it up. The sugars, that the pentose sugar, 5-carbon sugar, which have one oxygen in it, then 4 atoms of carbon in the ring. Just know that this one has one oxygen less, this one has one oxygen more, deoxy. So this should have 4 oxygen atoms, and this one should have 5 oxygen atoms, and hence it is called deoxyribonucleic. Then the ribose, the sugar there, is called deoxyribose. Then the sugar here is called ribose sugar. The difference is that this one have 5 oxygen, this one have 4 oxygen atoms. So from the sugar level, we will see the differences there. So we have then this is here. Here we have H and OH. OH. Okay, there will be OH. There will already be H here. That's the major difference. And H. And this one we have also OH. And this will have CH2O. So if we count these, that should be 1, 2, 3, 4, 1, 2, 3. These are only hydrogen. 1, 2, 3, and 4. Yes, so this is only hydrogen. There is no OH here. So this is the structure of deoxyribose sugar. Then that's the sugar in DNA. Then the sugar in RNA is almost just the same thing, but the difference is here. So over here we have OH, we have H, then here we have OH and the H. See where the difference lies. There is no O here, ordinary H is here. Then the same thing exists here, and we have H, then we have now CH2OH. So this one is the ribose, 5 oxygen atoms, then this is deoxyribose. So this is the sugar found in DNA, and this is the sugar found in RNA. At least that's one of their differences. But you should know now that DNA controls the RNA, DNA uses the 3 types of RNA in protein synthesis. The DNA makes use of messenger RNA, transfer RNA, and ribosomal RNA in the production of protein, in synthesizing protein. And remember, the expression of character is all about through protein synthesis. DNA have the information on a particular protein to be synthesized. Then it makes use of the messenger RNA in producing it. So what does this imply? That 2 of them collaborate during protein synthesis. The 2 collaborate during protein what? Synthesis. The only thing there is that the person with the plan on what to be built is DNA. Then with the help of the messenger, transfer, and ribosomal RNA, protein can be synthesized. What do we mean by protein here? Any biochemical substance in you, like melanin, is a protein. All the enzymes are proteins, and they are produced by joining amino acids. It is DNA that have the information on a particular amino acid to be joined. Then it uses the messenger RNA transfer and the ribosomal RNA in executing its function. This will be explained in detail when I discuss protein synthesis. That is the steps in protein synthesis in another video. So let's go back and focus on the biochemistry of nucleic acid. Actually, nucleic acids are polymers. They are actually polymers. When we say polymers, they are giant molecules. These nucleic acid, DNA and RNA are polymers. Where their monomer is called nucleotide. It's called nucleotide. What builds up both DNA and RNA is a nucleotide. The building unit or the monomer of nucleic acid is called nucleotide. The monomer of DNA and RNA is the nucleotide. So when you join many nucleotides, you have DNA and RNA. So the linking of nucleotide to another nucleotide gives you nucleic acid. So let's see now what are the components of a nucleotide. A nucleotide is divided into 3 main units. The 3 main parts. The 3 main parts of nucleotide. Are 1. A given nucleotide must have a pentose sugar. Pentose sugar, which is what I explained earlier, which can be deoxy, ribose, or ribose sugar. Then second unit of a nucleotide are the bases, known as nitrogenous bases. Then the number 3 unit is the phosphate group. Phosphate group or groups, depending on phosphate group or groups. So when you join 1, 2, 3, what you have is a nucleotide. Now, as we all know this, DNA is double-stranded, it's double helical, just like a twisted ladder. As you can see the structure on the screen. Then RNA have the shape of a half ladder. But from the structure now, you will now see which part represents the pentose sugar, which part represents the nitrogenous base, and which one represents the phosphate group. Once you just know this, number 1 and 3 forms the backbone, what we call sugar phosphate backbone. If you have a ladder in this way, number 1 and 3 forms the backbone. That's pentose sugar and phosphate group forms the backbone of DNA and RNA. In other words, we call it the phosphate sugar backbone or the sugar phosphate backbone. The sugar phosphate backbone. Which means if you come now, whereas number 2, please, number 2 forms the interior of the DNA and RNA. So DNA, to separate the twist, is in form of this. We have P, and here we have another. And continued. we want to continue it. So you can now see it's forming a shape, a ladder. So this is DNA structure, whereby the three parts I mentioned are represented here. Here is the sugar. This is the phosphate. Sorry, this is the deoxyribose sugar. Deoxyribose. Even here, the deoxyribose. Then over here, this part. This part is the nitrogenous base, or the base. Then here is the phosphate group. So what we mean here, if you circle out this now, this is just a nucleotide. This is now another nucleotide, because this is the base, the pentose sugar, then the phosphate group. As you can see, if you join this unit several times, you now have DNA, because we have this unit 1, 2, 3, 4, 5, 6. So you have 6 nucleotides here now, and that makes up now the DNA. If it is RNA, you only have but half. So this particular skeleton, the backbone, is what we call phosphate sugar backbone, or sugar phosphate backbone. Phosphate sugar backbone. That means phosphate and a sugar. Then the interior, the inside, is made up of the nitrogenous base. So if you come to DNA, the external frame is made up of the phosphate group and the base, the phosphate group and the pentose sugar, whereas the cross lines is now made up of the bases. The what? Bases. Then coming to bonds existing between each nucleotide. For a nucleotide to join another nucleotide, the bond that joins a nucleotide to another nucleotide is called phosphodiester bond. Phosphodiester bond. So the bond here now is phosphodiester bond. Here is phosphodiester bond. Here is phosphodiester bond. Then the bond between nitrogenous bases. Here are nitrogenous bases. The bond here is actually hydrogen bond. So the bond between phosphate sugar backbone, that's the bond between a phosphate group and a pentose sugar in the phosphate sugar backbone of nucleic acid is phosphodiester bond. So the bond here is actually called phosphodiester bond. Okay, this is just a brief explanation. We will take each unit and discuss in detail. So number one, pentose sugar. I've told you the difference is that from the structure, you refer to the structure I drew before mentioning it. You have the two types. Either we are talking about the deoxyribose or the ribose sugar. So that's just it. Please take this correction. Deoxyribose nucleic acid doesn't mean it doesn't have any oxygen. No. That's not the meaning. It's just trying to signify that it has one oxygen less than the corresponding RNA, as you can see in the structure I drew above. Maybe just for sake of clarity, see it very obvious. And we have another structure. This is what we call Franus structure. A Franus compound is a 5-membered cyclic heterocyclic compound made up of 1 oxygen and 4 hydrogen. So here is hydrogen number 1. Here is C1. Sorry, this is a carbon number 1, carbon number 2, number 3, number 4, then number 5. Carbon number 5 is here. The same thing that is applicable here. 1, 2, 3, 4, and 5. So you have the CH2OH here. So the only difference is that if you come to ribose, this is ribose sugar. You just have the hydrogen attached, the OH attached. But at carbon number 2, there is still OH and the H. Number 3, you just have but OH and the H. Number 4, you just have H and then CH2OH. But if you come here, carbon number 1 have OH, but number 2 have nothing but H. It lacks the OH. That's why it is called deoxyribose. They are all 5 carbon compounds. So if you come here, there is still H, there is OH, there is O here. So if you count this 1, 2, 3, and 4, but here it's 1, 2, 3, 4, 1, 2, 3, 4, and 5. So that's it. So this one is ribose, and here is deoxyribose. 1 oxygen less, 1 oxygen more. That is it for the number 1, pentose sugar. They are all 5 carbon sugar. Now, if you are asked what are the uses of pentose sugar, you say they are used for formation of nucleic acid. They are not used for production of energy, and they occur in Furano's structure. That is, they occur as a ring of 5 members, where 1 is oxygen and 4 are actually carbon. Okay, now let's go to number 2, nitrogenous bases. Cameraman, are you still focusing? Okay. Nitrogenous bases. The second unit of a nucleotide is called nitrogenous bases, or just the bases. These bases, nitrogenous bases, are divided into 2. There are 2 main groups of nitrogenous bases. They are purines and pyrimidines. Purines and pyrimidines. These are the 2 major groups of nitrogenous bases. Then, in nucleic acid, we have 2 common purines. There are only 2 purines that are common in nucleic acids, and they are the adenine and the guanine. Adenine and guanine are the 2 major common purines that occurs in the nucleic acid. Then this one, we have 3 main common ones that can occur in a nucleic acid. They are the cytosine, then you have the thiamine, and then you have the uracil. So the total number of nitrogenous bases that are possible are 1, 2, 3, 4, 5. There are 5 bases. 5 bases: adenine, guanine, cytosine, thiamine, and uracil. But out of these 5, no nucleic acid will have the 5. A given nucleic acid can only have but 4 out of the 5. A given, which means DNA have only 4 out of these 5. RNA has 4 out of these 5. To bring you to a faster way of understanding it, see what I mean? Out of these 3 pyrimidines, only 2 can be present in a given nucleic acid. No nucleic acid will possess all these 3 pyrimidines. But these 2 purines are constant in both RNA and DNA. Both RNA and DNA have adenine and guanine. But if you come to DNA, DNA have only cytosine and thiamine. Thiamine is only present in DNA. There is no uracil in DNA. DNA lacks uracil. So the nitrogenous bases in DNA includes nitrogenous bases in DNA includes 1, adenine, 2, guanine, 3, cytosine, 4, thiamine. Simple. These are the purines and these are the pyrimidines. So we have 3 pyrimidines, but out of the 3, only 2 is present. So it means that uracil is absent in DNA. Then, if you come here, nitrogenous bases in RNA includes 1, adenine, 2, guanine, 3, cytosine, then 4, uracil. which means in RNA, thiamine is replaced by uracil. Thiamine is replaced by uracil. Then in RNA, like when it comes to RNA, thiamine is taken over by uracil. But when it comes to DNA, uracil is replaced by thiamine. So no nucleic acid have both thiamine and uracil at the same time. At least you can now differentiate RNA from DNA based on their bases. You can differentiate them based on their pyrimidines. You can't differentiate them based on their purines. Their purines are the same. They are similar, adenine and guanine. But pyrimidines, they differ in their pyrimidine, which means that the pyrimidine present in DNA are cytosine and thiamine, while that of RNA is cytosine and uracil. So by so saying that 1, 2, 3 are constants for every nucleic acid, it is these two that varies. So this makes the difference between DNA and RNA. So now at this base level, we go detail in the biochemistry of these bases we are calling. First, when we say pyrimidines, what are actually pyrimidines? What are actually pyrimidines? We will see pyrimidine and see purines. What are pyrimidines? In organic chemistry, actually a pyrimidine is one of the isomers of the diazine or the azine or the azine. Diazine. Pyrimidine is one of the isomers of diazine. What is diazine? A diazine is a heterocyclic organic compound that is made up